All right, it's 3 o'clock. Let's start. And thank you all for joining. I'd like to welcome you to this workshop on multi-omics integration tools and platforms. This workshop is jointly organized as part of a series by BBMRI-NL, Eatris-Plus, HealthRI, and the Netherlands Cross-Omics Initiative. And for today, we invited 3 speakers who are going to present different platforms and toolboxes for multi-omics data integration. First, we'll have Remond Fijneman, who will present the cBioportal. Then we have Jan Costa, who will show us the R2 genomics analysis and visualization platform. And finally, we invited Kim-Anh Le Cao, who gives an overview of the mixOmics R package. But before we start, a few words on the agenda. So each speaker will present for about 20 minutes. And after each talk, there will be time for your questions. Please write your questions to the speakers in the chat. You can do this throughout the talks. And after each talk, we'll select the questions and we'll then ask you to unmute your microphone. Otherwise, we'd like to ask you to please mute your microphones. And with that, I'd like to welcome our first speaker, Remond Fijneman. He's a principal investigator at the Netherlands Cancer Institute in Amsterdam, where he works with the Translational Gastrointestinal Oncology Group at the Department of Pathology. His translational research focuses on biomarker identification and validation for colorectal cancer to improve, for example, diagnostic screening or disease monitoring, and to drive translation into clinical applications. Today, he's presenting the cBioportal, an open access platform for interactive exploration of multidimensional genomics datasets.
Thank you very much. Time-wise, please keep track of the time. Let me know when I'm approaching my end of the 20 minutes so that I can anticipate a little bit on the duration of the presentation. And let me... All right. Thanks again for the invitation to present cBioportal as a tool of, I think, much interest for the translational research that we are doing. But I think there's many applications and many people who could benefit from this tool. And I will talk about cBioportal. I will also talk about a use case, a study that we're doing. It's called MEDOCC. So in the presentation, what I would like to do in the beginning is to position the tool cBioportal in the translational research landscape. We are doing biomarker research, translational research meaning research that brings findings from the bench into the clinic, to the bedside. So basically what one tries to do is to translate, in this case, molecular pathogenesis of colorectal cancer into clinical applications of a molecular test. And this is, of course, a long and tedious procedure where one needs biomarker validation, assay development, prospective validation, clinical trials, etc. I think in cancer research there, and in particular in the biomarker research, in the end, there's only a few questions that are critical to address. Basically, one wants to know who has cancer, who needs additional treatment, how does this treatment look like, and when should one treat or stop treatment. When I later talk about the MEDOCC use case, it will actually address the question who to treat. So, when you look for biomarkers in the field of cancer, a logical biomarker to dive into is DNA. You know that cancer is caused by DNA alterations. And so when you search for DNA alterations, it makes sense to dive into tumor tissue because that's where the cancer cells are. But I will show you in a second that somewhat surprisingly, it's actually also possible to find DNA mutations that are derived from the cancer in liquid biopsies like blood samples. When you look at this schedule, what you see is actually that among the things we're doing is collecting patient cohorts that connects to these prospective validation clinical trials. One needs samples from these cohorts, like blood samples. One needs to do molecular profiling tricks, in this case, DNA profiling. And one needs to bring the data together. And that is the main topic of today's presentation, basically. Just very briefly, because I can imagine that this audience has quite a diverse background. So how come that DNA alterations from tumor tissue end up in the blood? That is because when cells die, apparently fragments of these cells do end up in the bloodstream, and that includes cell-free DNA. So in the blood from normal cells, you will find DNA fragments. Of course, that's the same for tumor cells. So when tumor cells die, also the tumor DNA to some extent will end up in the bloodstream. But because their cancer is characteristically different from normal based on the DNA mutations, it is these DNA mutations that you can also identify in the bloodstream. So that's called ctDNA, circulating tumor DNA, that we are after. When you do studies on patient cohorts, you want to do molecular profiling and you want to get access to the clinical data, think about the trials and the technologies, etc. Before you know it, you're working with a multidisciplinary team. And I show you here the team that many of the studies on ctDNA that we are doing. This is the team that is actively involved. And basically, just to show you the type of disciplines involved, there is medical oncologists and clinical trial specialists involved. The blood samples go to the clinical chemistry. The molecular profiling is done with assays that were invented by an expert in the field of ctDNA technology. In this team, I'm the molecular biologist. Then there is the pathologist, in this case, Gerrit Meijer, who also has a large role in the research IT infrastructure in the Netherlands. And we make use of the data for health technology assessment. So in this way, we work on a lot of studies, and this is just to show you that there's many of these cohorts that are being composed of stage 2, 3, and different types of metastatic cancer with hundreds of patients involved. Why is this important before I start talking about the tool cBioportal? It's important because one has to realize what actually happens to patients or study participants who enter the clinic or who enter a study. And then there's a number of steps that are taken. So on the one hand, there is the clinical information collected in the electronic health records, and that are collected in a database in which you will collect all this clinical information of your cohort. There's clinical imaging like CT scans, MRI, PET scans, but also digital pathology. And also here, you will have databases with the data for each of these imaging fields. Then there is the samples that you extract from the patients. So the blood samples, etc., that you store in the biobank. And you want to make use from these samples, the blood samples, the tissue samples, for instance, for your molecular profiling, DNA, RNA, protein, metabolites. And that's where you generate your experimental data. These somehow need to be brought together, maybe with the reference of external databases. And from here, you want to do the combined analysis, the downstream analysis, ending up with your papers, IP, but also with the main goals of your study, improve healthcare. And you can actually involve the participants in the patient advocates in this study. And when you look at a scheme, and I wonder where cross-omics, for many of the aspects of cross-omics is positioned, I expect that cross-omics, for a large extent, is positioned here in the experimental domain. The cBioportal tool tries to extract the cold data from each of the domains. So let's say the final results that come out of the molecular profiling, that are the results from the clinical information, from the imaging information and the biobank information. So this is where cBioportal is positioned. in this data integration part. Now, very briefly about cBioportal. It's an open access resource for interactive exploration of multidimensional cancer genomics datasets. The cBioportal significantly lowers the barriers between complex genomic data and cancer researchers who want rapid, intuitive, and high-quality access to molecular profiles and clinical attributes from large-scale cancer genomics projects. And I highlighted here actually this sentence, because I think there's a tendency that bioinformaticians are very strong in having access to the data, very willing to help out other molecular biologists, medical researchers who are not bioinformaticians. But in many cases, it's very nice to actually, as a non-bioinformatician, be able to already ask a number of simple questions to the data set that is now, without these kind of tools, very difficult. So I think here there really is a gap, a need that is filled by cBioportal. And cBioportal originates at the time from the Cancer Genome Atlas project. That is when the developers of cBioportal started to develop cBioportal with the purpose of accommodating data from the Cancer Genome Atlas. But if you now open the public instance, just go after this presentation, after the symposium today, to cBioportal.org, and you will find a long list of studies, and you can play around with all of these studies. So some are pan-cancer studies, many are cancer type specific, and it will give you a lot of insight of what cBioportal has to offer. And when you go there, one of the questions you may want to ask is, wouldn't it be nice to see my own study in cBioportal? One more boring slide on text slide on cBioportal. It was originally developed at Memorial Sloan Kettering Cancer Center in New York. And some of the developers that we are still in contact with, J.J. Gao, Niki Schultz. The public cBioportal is hosted by the Center for Molecular Oncology at Memorial Sloan Kettering. cBioportal software is available under an open source license via GitHub, so you can all download it if you like. And it's the software is developed and maintained by several, but a limited number of institutes, being Memorial Sloan Kettering, Dana-Farber Cancer Institute, Princess Margaret in Toronto, Children's in Philadelphia, the company The Hive in the Netherlands, and Bilkent University in Turkey. So, as I was saying, would it be nice to see my own study in cBioportal? I would love to. So let's go to MEDOCC use case. And as I already introduced, what's the question? The question is to treat or not to treat. If patients with a primary colon cancer undergo surgery, what is the chance they will develop a recurrence? Do they need chemotherapy, yes or no? The study that we run, we collect the patients by making use of the prospective Dutch colorectal cancer cohort. And that means that nationwide, patients are asked for informed consent to make use of their clinical data for research purposes. And these data, in the end up in the Netherlands Cancer Registry with IKNL. And then they're asked for permission for using tissue and blood and for, the patient's reported outcome measures. So inquiries about how patients feel. We collect blood samples before and longitudinally after surgery. Before surgery, we even do not know whether they're stage 2 or stage 3 patients. So we basically run multiple studies using this procedure. We collect the blood samples, ship them to Baltimore, for instance, to Jill Fallon in the lab of Victor Valculescu, who developed a technology called TAC-seq, and that allows for a very sensitive method to detect oncogene and tumor suppressor gene mutations in the plasma in a number of genes. Originally, we thought it's great to just look at plasma, but later we realized we should do the combination of tumor profiling, cell-free DNA plasma profiling, and to avoid being fooled by the white blood cells, in which there's often clonal hematopoiesis, in which there's often genes like DNMT3A, but also TP53 that are mutated. This as a background for the type of data we are collecting. Clinical data, Netherlands Cancer Registry, pathology data, Impalga, digital pathology coming up using a tool developed by Jan Hudecek, molecular profiling by the colleagues in Baltimore, but also by now by a commercial company. And all of these we try to bring together in cBioportal. cBioportal, as I said, the public instance is hosted in New York, but in the Netherlands, we have an instance that's hosted by HealthRI. And HealthRI is basically an umbrella organization under which TRADE, a former project that at the time I was actively involved in, but also cBiomics and a lot of other infrastructure projects in the Netherlands. So if you go to the HealthRI website, you can go to cBioportal, and then here it states cBioportal, and you can click on it, and then you get access to your instance and your studies if you sign in using your Google credentials. Initially, this is very scary because who wants to put their studies with Google? That doesn't sound GDPR proof, right? But really, this is just using the credentials. It's not that you are referred to Google. The other disclaimer I want to make is that MEDOCC is really a study that is in progress. So the data that I'm going to show is still, well, some of the details still need to be filled in. But it also shows that it's actually possible that while you're working on a study, you can already enter part of your data in cBioportal. So when I log in, here I see my studies, and that really makes me happy. And then I can click the one I want to show you today, so the MEDOCC study, and I will explore the selected study. And then I open it, and it's like I'm looking at the dashboard of the COVID update. In this case, it's the dashboard of my study, and I see a little bit of a survival curve. I see what genes that we identified were frequently mutated. I see the distribution of genders, so sometimes in the pie diagrams, sometimes in tables. You can change that if you want from pie diagram to table, etc. There's these bar plots, and you can actually select the younger or the older patients if you want. So there's lots of selections you can make. And there's lots of options that you can see in your dashboard. And basically all the data that you collected are found under this table here. And whenever you click, it will fill in the dashboard. So these filtering steps are really useful. And in this case, there's lots of data in it, but I'm actually only interested in the samples where we did some mutation profiling. So that is what is now selected here. And then next to the summary tab, there is the clinical data tab. And now you're entering basically a spreadsheet that is very much the same like an SPSS spreadsheet or like an Excel sheet. And so here you see all the data that you would find in such a spreadsheet. And if you want to do downstream analysis that cannot be done within cBioportal, then you can simply download the data and fill your spreadsheet, your own spreadsheet, and do all other analysis that you like to do. So that was on the study level, but of course it's also important to see what happens to individual patients. So here apparently with the selection we have 158 patients for which there's information. Now I click on this button over here, and then I see this longitudinal view of a single patient. And the reason that I emphasize longitudinal is that in most tools, the possibility to actually visualize longitudinal data is lacking. And that is, of course, quite critical for a lot of studies. So here you see the patient timeline, time of diagnosis, then there's this blood sample taken. The red ball is the ctDNA, the white one from the same tube, the white blood cells for clonal hematopoiesis, surgery over here, tumor being collected, the tissue, and then another blood sample, and apparently this patient received chemotherapy for this period in time. When we move down a little bit, we see these little balls again popping up, listed per gene mutation. So apparently there's an APC mutation present in the tissue, observed in the pre-surgery blood sample as well as in the post-surgery blood sample. Likewise for these others here. And then you can also see alterations like DNMT3A present in one of the blood samples, but also in the white blood cells. And that shows immediately the warning sign, now, ooh, that's probably clonal hematopoiesis and not a somatic mutation in the tumor. We can also go again to the clinical data tab and see all the information for this individual patient. Then we can go to the per gene level, for instance. So here I selected only the tissue samples to dive into, and I requested 4 of the common oncogene suppressor genes that are mutated, APC, P53, KRAS, PI3 kinase, also listed over here. And then you can see something that's called an oncoprint. So every block here vertically is a single tumor sample, and you can see whether or not there's a mutation in that particular sample in APC, P53, KRAS, PI3 kinase, whatever genes you selected. You can add clinical tracks. In this case, I added gender and mutation count on top of them. And so you get a very comfortable overview of how these oncogene suppressor genes are present in the same samples or are mutually exclusive. There's also statistics that one can apply on that, etc. You can also go to the individual gene. In this case, I selected the APC tumor suppressor gene, and in this lollipop figure, you can immediately see where these mutations are, in APC quite dispersed, and in what samples they were present. And then you can also do group comparisons if you want. So I selected here already a group of KRAS mutants, and I'm doing here the same for P53 mutations. So you create groups, and then you say compare. And then you can get something like this, where you see, okay, there is these 2 groups, they're overlapping, but in the comparison that will be shown, it will not include the ones that are overlapping. And you can say, oh, let me show the survival curve for the KRAS mutant only and for the P53 mutant only, and you get something like this. If you're interested in cBioportal, having seen these slides, and if you haven't been there before, please go to the international server and have some fun with some of the studies that are there. But for the studies in the Netherlands, please consider to make use of the HealthRI instance of cBioportal so that there's authorized access for your consortium to make use of cBioportal to accommodate your translational research. And if you want to do that, you can simply mail to the service desk at HealthRI. And there's online tutorials, etc. A few more slides to end with, future perspectives. So some of the developments that are ongoing at the moment is that there's integration with digital pathology. I mentioned briefly Jan Hudecek. He is involved by HealthRI and also in our institute as a cBioportal expert, but he also created a tool called SlideScore, which is excellently suited for digital pathology. So he's working together with the cBioportal team in the US to have this additional tab in the cBioportal tool where one can connect to SlideScore as digital pathology. And while they're doing that, they're actually doing the same thing for the clinical imaging. So basically one can do the same thing for CT scans, MRI scans to connect to them as well. And the other thing that's being worked on is you saw the mutation profiles on the tumor tissue. We are doing ctDNA research in a longitudinal manner. So basically you would like to see the profile of the mutant allele fractions from the ctDNA in time in the patient. So the plot like this is, well, a first plot that is being created, but could be a component they want to add to cBioportal. The reason that I show you this is that this tool is still expanding, the user community is expanding, but also the functionality is still expanding. Then I have future perspectives with a few question marks. So we're doing translational research. One emphasizes then multi-center consortia, But what is really needed is more automation of data flows. How can you more easily transfer data, clinical data, once they have been entered in a system, into a tool like cBioportal for translational research purposes? So for instance, the clinical data from the Netherlands Cancer Registry, why can't they just be automatically pushed through to a tool like this? Same for the pathology data in Palga. Same for the laboratory management systems in each of our institutes for the biobanking data. And the molecular data, you know, we're doing now molecular diagnostics. So we are moving away from a laboratory setting, a research setting only, to pipelines for DNA analysis and other analysis that are quite frequently used. And those data could also, should also be automated for easy entrance into a tool like cBioportal for translational research purposes. But not only for translational research purposes, also for the local needs for the medical doctors, you could do this. You could think about cBioportal actually as a tool to have a simple overview, a multidisciplinary overview of the status of a patient. So you should consider to use it for the molecular tumor board and as a tool in the multidisciplinary meetings when patients are discussed. And then, of course, you do need local instances of cBioportals in the individual hospitals, because then it becomes part of the hospital system, in which case it becomes attractive to think about federated data access to the different local cBioportals. And last but not least, publications. If I open the New England Journal of Medicine, I'm always very much annoyed by the beautiful studies that are being described and the painful way of not having access to the data behind it. Wouldn't it be great to publish in the New England Journal of Medicine and then actually say, and this is the cBioportal instance of the study. So you can go there and actually ask your questions on the trial that we published. Last but not least, acknowledgements. So the person driving a lot of the research IT infrastructure in our group, the head of the group, but also for the CTMM projects and also as a founder of Health.RI is Gerrit Meijer. And I found this slide where he's put on a slide and it says, tools themselves are not all that important, meaning that in the end, it's more important to have fair data, findable, accessible, interoperable, reusable data, than to have fancy tools. But it does help to have a fancy tool like cBioportal. And I think it's actually fantastic that cBioportal is around. Gerrit is not alone. He has been helped from the start of trade by Mariska Bierkens. So she really is Gerrit's right hand in terms of research IT infrastructure. I have been traveling with Mariska to New York, to Boston, to the people behind cBioportal. And this is actually a picture taken on our way back in a tube station called Government Center. But I think together they are actually the government center of a lot of the directions that research IT infrastructure should take, together with Jan-Willem Boyte and Rita Azevedo in the past from trade, ligature, and now Health.RI, where the data team who is contributing to cBioportal is expanding with Lana Meikari, Menno de Vries, Brenda Heijmans, Noriko Kasman, and Jan Hudacek. With the studies that we do based on the prospective Dutch colorectal cancer cohort, with the program manager Geraldine Vink, and last but not least, directly with the people from cBioportal, in particular JJ Gao and Nikki Schultz. But it turns out that there's actually a Dutch person, a software engineer working at cBioportal team in New York, who is now doing his PhD together with us, meaning that there's actually quite some active interactions with the cBioportal community. With that, thank you for your attention. I'm happy to take any questions.
Thank you very much for this insightful overview of the cBioportal. I don't see any questions in the chat yet. Okay, so there is one from Mark Hellings. Can you unmute yourself to ask the questions? The question, Martje?
Hey, Remond, thank you. A very interesting talk. I was interested in how do you add your clinical and genetic information into the database? So what kind of format do you use? Is this, for example, automated or in a standard setup?
Yeah, so I'm the molecular biologist not entering the data myself. That partly answers your question and partly it doesn't. So there is a fixed format of entering the data. And of course, that means that the molecular data, but basically any data type, have to, how do you say, comply with the data formats that is prescribed. You can find that information on the information websites for cBioportal. I, as a molecular biologist, am not entering the data in there. So we do have people who are experienced in doing that. It's not extremely difficult, but it's also not meant like I do an experiment today and I just enter it on a daily basis from those experiments into cBioportal. Yeah, but I'm not sure whether I really answer your question, Maartje.
Well, I think I should just check the website out myself and see if I can figure it out. Thanks.
And if you have questions and if you need help, then please go to the service desk mailing address and or send me a mail that I can bring you into contact with our people who are doing the upload of the data in cBioportal to get you quickly connected to the right information.
Great, thanks. Definitely will do.
Thank you. And there is another question from Anna. The question was whether it's possible to collect data on oral cancer in the cBioportal.
On oral cancer? Yeah. On any cancer. I mean, you can, so if you have your own study, you can collect and you can look in the public data whether it's already present or not. But basically, you can collect data for any cancer type.
Okay, so I could collect my own data, like I could collect data from that database, right?
You want to collect it from the database?
That's also possible. You can go to the public instances of cBioportal and download the information that is in there.
Thank you for the question. I have a final question. When you use the HealthVRI hosted instance of cBioportal, is there, or how does it work if you would want to share data across studies? Is there a data access committee in place so that you could request studies from other researchers?
Yes, so when you have your own study that you want to host with HealthVRI, you can apply for that. And then you can indicate who you think should have access to that study. Now, when someone else has the question, can I access your study? Then they will mail HealthVRI and HealthVRI will mail the PI of the study and say, this is a person who requests access. Do you approve or do you not approve? And so what happens in practice is while you are building the study and it is not published and it's work in progress, like the MEDOCC use case I showed today, this is not open. So it will be restricted to the consortium members. Those will have access, no one else. But by the time that there is requests from others to have access, then that request in this case would reach me as one of the PIs of the study. And then we can approve or deny and have our mechanisms in place in terms of a data contract if needed. So basically the study owner is in control of who has access.
Okay, thank you very much. And there are no more questions. So I'd like to welcome our next speaker, which is Jan Costa. He's being trained as a molecular biology and cell biologist at Utrecht University and at the Netherlands Cancer Institute, and switched to the then emerging field of bioinformatics in 2003. And since 2007, he's a team leader of an applied bioinformatics group at Amsterdam UMC. And his group developed the R2 platform to enable researchers to analyze and interpret their own high throughput molecular data. And today he'll give us some insight into the development of this platform and its use in research. With that, I'd like to come to our final speaker today, which is Kim-Anh Le Cao. So after graduating from her PhD in statistics in 2008 at the University of Toulouse in France, She soon moved to Australia to work on biomedical problems, and she's now an associate professor in statistical genomics at the School of Mathematics and Statistics at the University of Melbourne. And her research focuses on multivariate statistical methods for data analysis and integration with application for microbiome, single cell, and omics data. And since 2009, her team has been working on developing the R toolkit mixOmics that is dedicated to the integrative analysis of omics data. And today she's giving a brief overview of the main concepts and methods implemented in mixOmics. And due to our time difference with Australia, we recorded this talk before, but Kim-Anh is available for answering any questions you have on her talk, which you can ask in the mixOmics user forum, and we'll share the link.
So it's my great pleasure to present to you some of the methods we have implemented in mixOmics for multi-omics data integration. So the area we work in is between biology and statistics. So biology tends to be more qualitative, and statistics and mathematics tend to be a bit more quantitative. And it's really our job, when we have developed those methods, to try to bridge the gap between those two disciplines. This requires a lot of collaborations and tight interactions between different players. And the reason being that we have to face a lot of challenges. The first one is that we have a lot of data that are coming from multiple sources and multiple platforms and technologies. We also face computational issues because those data sets are very big and it's really hard to make sense of those data. The sample size is usually very small, and so that means that it is very difficult to validate the results statistically. And so instead, we have to rely a lot on the biological interpretation to validate the meaningfulness, how meaningful those results are. And the final challenge we face is that we have to constantly keep a pace with new technologies. For example, now we have single cells platforms and technologies that rise, and they're looking, for example, at spatial information of the cells as well as the transcriptomics information. So what is the systems biology trying to do is trying to understand how a biological system works in a holistic way. So if you remember 15 years ago, for some of you, we used to have a very reductionist approach in biology, where we would have a hypothesis on a given gene. We would knock out that gene and do an experiment on that gene, and then we would conduct one statistical test. But with the omics assays, we're measuring thousands of molecules at once. And so it is very difficult to understand even what the hypothesis might be there that we want to test. And when I talk about omics, I talk about transcriptomics. So that's the study of all the transcripts that we can measure in a biological system. Proteomics, metabolomics, microbiome, epigenomics, any big data basically that you can measure to try to characterize a biological system. And the relationship between these omics is not straightforward. It's not a linear relationship. And so that makes any analysis, statistical analysis, actually difficult as well. And so we started from a reductionist type of approach, where we would look at a single RNA molecule across individuals. We went then a few years later to single omics, where we would measure thousands of the same type of molecule across individuals, for example, here in transcriptomics. And now we're entering the multi-omics era, where we are measuring different types of omics across the same individuals. And here we're trying to really understand what are the interactions and the relationship between all these different molecular layers. But it doesn't mean that we have to completely rule out the reductionist type of approach. In fact, what we're trying to do with a multi-omics analysis is generate hypotheses that we want to test back in the lab. So how can we reduce this amount of information into just a subset of genes or proteins that we can then test back in the lab? And there are different types of methods for data integration, and I'm sure you've heard about some of them. So matrix factorization techniques is what we focus on in mixOmics, but we also have Bayesian approaches that have been proposed, network-based approaches, or also multi-step approaches. So those approaches, the multi-steps one, really look at analyzing each single omic layer at a time, identify what are the key markers in each of these layers, and then look at the overlap, for example, at the gene level between those results. So I will give you an example of such analysis in the case study. So what we're trying to do with our methods is really to shift this one gene hypothesis paradigm, because we believe that molecules act together to trigger cells' response in biological system. And so in multivariate analysis, what we do here is we propose methods that can identify a combination of markers rather than considering each univariate marker at a time. And those methods are also helpful to reduce the dimension of the data. And so they give us more insight into what is happening in the biological system. And you will also see that they are amenable to data integration, or the integration of different data sets that are measured on the same individuals. But before you go into that field, I think you really need to ask yourself the question, what is it exactly that you would like to get from this kind of analysis? Some researchers might be interested in understanding the role of each omics in the biological system, or trying to understand the relationship between these different omics types. Some researchers might be interested in identifying a molecular signature that really characterizes that system. Or they might be also interested to see whether they can predict the outcome of patients based on all these omics. And of course, you might also be interested to answer any of those questions above. So matrix factorization techniques here, I'm just gonna give you a brief introduction. What they're trying to do is they build artificial variables that we call components. And those components aggregate the original variables, for example, the genes or the gene expression or the transcript or the protein abundance. And those components are built so that they summarize the information from the data. And usually that information is based on either variance or covariance, depending if you're looking at one data set or two data sets or more than two data sets. And they're very useful because those components reduce the dimension of the data. So instead of considering a big data set, here you have n individuals or samples and p features or p variables. We're going to only summarize this big data set into a set of components. What is also interesting with those methods is that they are non-parametric. They don't make any assumption of the distribution of the data. And they're exploratory. So we don't require a very large number of samples in order to explore those data. And so examples of those methods are principal component analysis, but I will focus more on projection to latent structures or PLS models because they're very useful for data integration. So I'll start first briefly with PCA. So here what we're doing is we're trying to build those components, and each component here is a combination of the original variables that we have here. And the way we combine those variables is we assign them a weight. So the weight here is indicated in another vector called a loading vector. And that loading vector really tells us how important each of these variables are to build the component. And the component should maximize the variance or the information from the data. And so this is how you can then summarize a very big data set into only a set of a few components, because those components are supposed to maximize the variance from the data. And the other variation or variant that you can have from this method is to do feature selection. So you use lasso penalization in those methods, so that some of the weights in the loading vector are going to be set to 0 because some of the genes or the features are not considered as important to explain the variance in the data. And so the components are going to be based only on a very small subset of genes, while still maximizing the variance in the data. So this is just PCA, but you can then extend the same concept for more sophisticated problem. For example, in discriminant analysis, what you're trying to do is you're trying to discriminate different groups of patients here. So here they're indicated by 1, 2, or 3. And so you're trying to find or identify components that explain the differences between the sample groups. And so the components and the loading vectors are going to be different from a PCA, because here we're really trying to maximize the differences between groups. And again, you can do also variable selection or feature selection, where you can identify what is a set of genes that are key discriminants and help you to separate those groups. And so we then extract those components here, and we can plot those components basically in a scatter plot. And so this is what you can see here on the same data set. So it's a data set with 63 samples. We have about 3,000 genes. And here you have, on the x-axis, you have the first component, and on the y-axis, the second component. And the samples are represented as dots here in that plot. What it means really is that if you plot those components, you're actually projecting a very large data set into a space that is spanned by only two dimensions or 2 components. And so when you do PCA, this is an unsupervised technique. So really you're looking at clusters of samples, because once they cluster together, it means that they are similar in terms of their gene expression. But with discriminant analysis, you can see now that there's a coloring that appears with symbols, and really you're trying to discriminate the different groups. Here they are different types of cancer. So you kind of constraining the analysis so that the components really try as best as they can to discriminate those sample groups. And the latest approach that we have developed is to integrate more than one, or to integrate data sets. And so the idea is the same. It's just that here I changed a little bit the layout. You have different data sets. They're measured on the same samples, but they are using different omics technologies. For example, you may have transcripts, proteins, metabolites. You can also have an outcome, so you can do a supervised analysis. And the idea is the same. You want to decompose each data matrix into a set of components and associated loading vectors. But there's an additional constraint here that you want to maximize the covariance between the components that are associated to each data set. So what that means is that you're really trying to extract common information between those data sets, only the common information between those data sets. And you're trying to basically identify subset of genes that are correlated with subset of proteins that are correlated with subset of metabolites that also explain an outcome. So this is our latest multi-omics method that we have developed here. And then it's the same. You can extract those components and visualize them in a sample plot. So I'll show you an example here. So just remember, for each data set, you have a set of components, but you need to remember that those components are supposed to explain the same thing across all data sets. So this is an example from a breast cancer study from TCGA. And you have the components here, and the dots and the colors here represent different breast cancer subtypes. And you can see here, for example, that the mRNA dataset seem to discriminate the basal subtype really well. Whereas if you look, for example, maybe at the microRNA or at the protein, it's a bit harder. So this kind of plot really tells you what is the discriminative capacity of each of those data sets when you try to integrate them. And then I told you that you can also do feature selection. So we have created different types of plots. This is a circus plot. And basically it shows on the quadrant here, it shows the mRNA that were selected by the method, the proteins that were selected by the method, and the microRNA. And you have a line that is either black or brown to just show the type of correlation, whether it's a positive or a negative correlation between those omics. And so based on that, you can quickly visualize, whether microRNA and mRNA tend to be more positively correlated. There's more positive correlation than negative correlation. We can see here that mRNA doesn't correlate very well, or it correlates only poorly with proteins, but proteins might tend to be more correlated with microRNA, just as an example. The other type of variable plot that we show in the package are correlation circle plot, and those kind of plot originate from PCA, but they were extended for multi-omics integration. And so here, each symbol here represents a specific variable that was selected. And then when they cluster together, it shows that they are highly correlated. When they are on the opposite side, it shows that they are negatively correlated. And of course, you could change that plot to actually show the identifier of the variable that was selected by the method. So this is just to give you a brief overlook of the type of outputs you get. What you need to remember is that those methods reduce the dimension of the data by decomposing the original data matrices into a set of components and a set of loading vectors. The components tell us how similar the samples are based on the features of the variables. And the loading vectors tells us which variables are actually important to maximize the correlation between those data sets. So moving on now, I'm just going to show you quickly one example where we applied Diablo. So this is called a small big study. It's a small study in the sense that we had to start from a very small biosample amount. It's less than 1 milliliter of blood in newborns. And it's big because we had 5 data types. So we had sample blood from at different times during at birth, and then up to 1 week. And then we have measured the transcriptomics, proteomics, metabolomics, and some cytokines as well. The strength of this study is that we have actually 2 cohorts. We have one cohort from Gambia, and we use that cohort to train the Diablo model. And we also had a validation cohort from Papua New Guinea, where we could actually do some prediction and test the performance of that model. So here I'm just going to focus on the training only, but if you go back to that paper, you can also see how we validated the results on that other cohort. So if you start with a single omics analysis, and I think you should, basically what we saw here is so here you have the number of transcripts that are differentially expressed compared to day 0. So at day 1, there's not many transcripts that are expressed, and as time goes on, the number of transcripts that are differentially expressed compared to day 0 increases, whether they are up or down regulated. And it's the same for proteins, and it's the same for metabolites. So we see that there are some changes. And so one multi-step analysis is then to combine those results and look at the correlation between those transcripts, proteins, and metabolites that are differentially expressed. And what you can see is not much, actually, because you have all these hairballs that shows that everything seems to be correlated, but you can't really work out why. And so this is just to summarize that the first step, or the first analysis that we did, is basically you calculate the Pearson or the Spearman correlation between the differentially expressed features. And you can see here from that heat map that the correlation is not very well structured. And so when you show it as more like a network-based visualization, it just shows that hairball. But if you do if you apply a multi-omics integration method, where the aim is really to disentangle what are the features that are correlated together, while also explaining the outcome. So here the outcome was the day of sampling. Then you can see a much better structure in terms of the correlation between those features. And what we found out is that each component identified by the method, by Diablo, and the features that were selected in those components focused on very specific pathways related to immune system, as well as cytokine signaling. So we obtain new insights by doing this multi-omics integration. We identified specific genes that we hadn't found before, but we also find common pathway information that we also had found in the single omics analysis. So a multi-omic analysis is going to complement basically the single omic analysis. And so I've been talking about mixomics. I've been talking about, three different approaches, PCA, the discriminant analysis, and Diablo, but in fact, there are many more methods in that package. One that I haven't touched on is the integration of independent data sets. So for example, transcriptomics data set that are focusing, or transcriptomic studies that are focusing on the same biological question or the same disease, but they have been generated in different labs, and you're trying to identify what is the common information you can extract from all these independent data sets. So that's MINT. And for those of you who have used the package before, you know that the sample plots are the same, regardless of the method you use, and similarly for the variable plots. What is important when you try to analyze those data is you really need to ask yourself, what is the biological question you're trying to answer? Because depending on the biological question you're going to ask, you're going to apply a very specific method. For example, you might be interested in only analyzing one data set and you want to do a supervised analysis. You don't want to do a regression. Or you may want to integrate two data sets and you want to do a discriminant analysis and so on. So it's very important that before you jump in multi-omics integration, that you ask yourself, if you can break it down into different biological questions and then apply different methods to answer those questions. You will see our latest updates. They're usually on our website, mixomics.org. There's going to be a book that's going to be published hopefully in a few months. And we hope to also advertise some online courses by the end of this year. So if you want to go further, there's a lot of extensions that we've been working on. For example, we looked at the integration of microbiome and omics measured on the microbiome, so meta-omics. And for that, we applied Diablo, so you can have a look at that paper. We've been also working on developing methods to integrate time course omics data and try to extract coordinated profiles across time. I haven't told you, but in Diablo, there's also a way where you can include pathway information in the analysis. And we're also focusing a lot of our work in microbiome to remove batch effects. So you can have a look at that manuscript in bioRxiv. And we're also looking at, I haven't put it here, but at the integration of multi-omics in single cells. Those are my conclusions. As you know, we're entering a very exciting era where we rely on data-driven approaches first to make sense of big data. And this is mostly to generate new hypotheses. It doesn't mean that we have to exclude a priori biological knowledge. You are still able to include that in those kind of methods. But for me, it's a very exciting field because it blends very different disciplines from computing, statistics, and biology. And as you know, the development of those tools is really crucial also to advance knowledge. And so I'd just like to finish on that slide to acknowledge some of the people who've been working with me, not all of them, but mainly the mixOmics team and all the users who have been helping us throughout to develop and improve the package. People from my team, as well as many other collaborators in different application fields and also different methodological fields who've been contributing also to the development of those methods. Thank you. And I'm also very happy to take any question. We have a discussion forum dedicated to mixOmics. You will find this on our website and we will share the link. And so feel free to ask any question there.
Thank you very much, Kim-Anh, for this really interesting overview of the mixOmics package and especially its role in the hypothesis generation. So I'm not sure if we have time for many questions, but I have a few questions. So one is, you mentioned that you use especially matrix factorization methods for hypothesis generation, and then you would go back to the lab to test these hypotheses, but that you also have the possibility to integrate pathway knowledge in mixomics itself. So do you have examples where such an approach has been used? I saw there is a, sorry,
feedback. We used it in one asthma study here. And so basically what we did is we first grouped the genes and the metabolites into pathway modules that were known. And then we summarized that information at the pathway level. And so that it's almost a way of already reducing the size of the data set. And then what you do from there is that you work you then integrate those different pathways rather than individual genes for example, in the Diablo method. So that's one way. The other way that is more classic is to just, do the analysis that I showed you before, and then you look at the pathways of the genes or the proteins or the metabolites that you have identified, and then try to see, if those pathways make sense.
Yeah, thanks. And I have another question regarding the supervised analysis. So you showed the example of Diablo, that it could be used for supervised analysis with a single outcome variable. Is there a method implemented that would be able to deal with multivariate outcome variables? How would you approach this?
So you mean by multivariate outcomes, you mean different vectors?
Yeah, like outcomes? If it's a matrix instead of a vector. Yeah, I'm thinking of readouts of variables or extensive phenotype information or exposure information.
I think it would be a bit difficult because really you can try, we definitely run, but I think the interpretation would be difficult because if you think about how many classes or how many groups it represents, it's a lot because there's all this combination of all these different variables. So I think it's more in terms of interpretation that it would be quite challenging.
And the power would be lower.
Yeah, although, with these methods, we don't even talk about power.
Yeah.
Because they're not inferential methods here. There's no p-values, so they're very exploratory. But I think it's more a matter. I think your question is more about, can you decompose all these data, this signal into chunks that would explain different outcomes at once? And I think that's a very difficult question.
Yeah.
I have a small question. Thank you for the presentation first. Yeah. You also showed about the first, I didn't know about that you can include actually time series data.
Yeah.
And but in one of the example, you also showed it actually that when you integrate 4 different omics types, that one of them were explaining at least one category or one class very well. So is there also an option once you start to integrate all these different omics types that it says, okay, this omics have more explanatory power or more explanatory so that the data information so that it can explain better. So it can categorize. So for example, microRNA explains less and or
yes. Yes, so from the output, and especially in a supervised framework, it's very easy to assess the classification performance. And so we do a lot of cross-validation, and the cross-validation and the prediction are based on the components. So you from the outputs, we give you also the error rate that you can, the classification performance per data set, because we extract each component from each data set. So you can have a result that is, not only visual, where you can see that, the method is struggling, but you can also have an output that tells you numerically that you may have a data set that is a bit weak for the discrimination. And of course, that will, if you're interested only in classification, mainly in classification, if you have a weak data set, it's really going to impact because it brings noise in the system. And so it will also, affect the classification performance. But what was your question about time course then?
No, I thought I told that I didn't know about that. You can actually integrate the different time series.
Yes, it's a very it's a very challenging question to look at time course, multi-omics time course data, and one reason being that there's some time lag often that is expected between different omics layers. There's a lot of noise because you have a very strong individual variation here. You can imagine that even if you have a very specific intervention that you're doing, some patients will react very differently, even if they have the same intervention. And you can see that at the molecule level. And so you have to look at time lag, and I think this is where the field is going now. There's a lot of time course data people are generating a lot of time course data. And we're still scratching our head on how we can actually solve or answer the questions.
Well, the reason also asking of a different explanatory value of different omics data set, you already mentioned that some of them would add also a lot of noise. So if I have, let's say, 5 different omics data sets, but when I integrate only 3 different combinations of it, gives me much more power, much more predictive power compared to the all 5 combined, then that would be very nice to know it.
I think it really depends on your aim. If your aim is really to classify, then you need to be ruthless and just focus on the omics that you think have some value. If your aim is more to understand a biological system, then it doesn't really matter, or it does matter, but not too much, because you really want to include all this omics information to understand at different levels, different molecular layers, what impacts your system. So that's why I was saying, the questions you need to ask first before you even embark on data integration need to be quite pointy.
Okay, we have to wrap up. So thank you again for the presentation, and also for being available for questions from the audience. Thanks.
Thank you.
Okay.
Thank you.
All right, so that was the recorded presentation from Kim-Anh Le Cao. Please don't hesitate to contact her if you have any questions on her talk or on the mixOmics package. We shared the link to the user forum in the chat. And before we come to the end of this workshop, we also invite you to join our next workshop, which will highlight predictive multi-omics biomarker signatures It's going to take place in 2 weeks from now on February 24th. And if you have any questions on the workshop series or on the CrossOmics initiative, you can contact the CrossOmics help desk. We'll also share the contact in the chat. And finally, I'd like to thank all speakers and also the CrossOmics project management team for making this workshop possible. Thank you. And also thank you all for joining and have a good day.